Well, good evening and welcome everyone to the President's Lecture Series. This is part of the Shepherd University Lifelong Learning Program under the outstanding leadership of Karen Rice. Karen, where are you? Yeah, right here. Thank you so much for everything you do for Shepherd, for our community. Well, we are just thrilled this evening to have the opportunity to host Dr. Juanita Anders. She's going to be lecturing to us on photobiomodulation therapy. What is it? How does it work? And what exactly it can do for all of us. We're going to be experts in this area by the time this lecture is over. I know it. Uh, yeah, Dr. Anders is internationally recognized as an expert in photobiomodulation, which we refer to as PBM. And in her research, she has served in noteworthy, prestigious positions. PBM is the biological response to using non-thermal, low-level light therapy to heal a wide range of injuries and diseases. Her specialty is peripheral and central nervous system injury and repair mechanisms, as well as light tissue interactions. Dr. Anders received her Ph.D. in anatomy from the University of Maryland Medical School and then joined the National Institutes of Health in the Laboratory of Neuropathology and Neuroanatomical Sciences, known as NINDS. She is affiliated with the Uniformed Services University Health Sciences as a professor of anatomy, physiology, and genetics, and professor of neuroscience. Dr. Anders serves on the executive councils and scientific advisory boards of numerous international laser conferences. She is the past president of the North American Association of Laser Therapy, a founding member of the International Academy of Laser Medicine and Surgery, and past president of the American Society of Lasers in Medicine and Surgery. She has recently been appointed as a board member of the International Society of Lasers in Medicine and Surgery and currently serves as director of the Optical Society of America Photobiomodulation Technical Group. She is a senior editor of Photomedicine and Laser Surgery, associate editor of Lasers in Surgery and Medicine, associate editor of Lasers in Medical Science, and on the editorial board of Physiotherapy Practice and Research. She has published over 70 peer-reviewed articles, and we are so fortunate to have this pioneer in this area of research come to Shepherd University. She is also an adjunct faculty member at Shepherd. I just want to point this out. We're very proud of that. We're very grateful that she took the time out of her busy schedule to join us. And then I would just like to invite everyone to join us after the lecture this evening for a Shepherd University Foundation sponsored wonderful reception in the lobby. So thank you again, Dr. Anders, for joining us. Thank you.
Well, that was a wonderful introduction, and it's so wonderful to see so many people here this evening and interested in photobiomodulation. This is a great honor to be asked, and I hope that I make you happy in the next the next hour. So, let me get started here. Besides thanking Dr. Hendrickson, Professor Hendricks, or President Hendricks, all of those names, besides thanking her for her efforts in photobiomodulation, among everything else she does, is I really do want to thank the community, staff at this university who have really made it a goal and have worked hard for the last, I think it's about 2 years now, with this vision of creating in this region an excellent center of photobiomodulation that would be a leader in education in this field and developing practitioners in this field, and hopefully of really building the research efforts here at Shepherd and in the surrounding area And I want to thank all of you who have seemed to be really interested in this potential therapy and who have come out tonight to hear about what it can do. I'm just so proud to be included in this effort. So I am a professor at Uniformed Services University, and that's in Bethesda, Maryland, on what was the Navy base, but now we're the new Walter Reed Medical Center, if you've gone there. And because that does belong to the Department of Defense and we train doctors for the military, we like to really think of ourselves as America's medical school. And if you'd like to know anything about that after this lecture, I could tell you about the wonderful opportunities there for you to serve your country. But I am a DoD scientist, Department of Defense scientist, and I must tell you that the opinions expressed this evening by myself are not those representative of the United States Department of Defense, the Uniformed Services University, or the United States Army, Navy, and Air Force. So if you have any problems with this talk, my email will be at the end. Please write me and not the president of my university. So I promised in the title that I would tell you exactly what photobiomodulation therapy is. But I'd like to start out by telling you what it isn't. Because if you go online, I think it's extraordinarily confusing on all the types of photon, or in a very general sense, light therapy applications there are. When I first started in this field, after I had been awarded a grant in Ronald Reagan's Star War program, I didn't know anything about this field. And at that time, it was referred to by a lot of different names, including low-level laser therapy. But I started going to meetings, and there were clinicians who said, and this was basically at that time a red light device, this device, red light pointer, will cure everything. It's magic the way it works. And that did not do this field any favors. People don't want magic. They want grounded, scientifically proven modalities. The other thing, as I mentioned, if you go online, you're going to see a lot of things about rays of healing light. And it's sort of mystical, divine interception of light. Or things like energy sources that repair the chakras or energy centers of your body. This is not that either. Also, when I talk about photobiomodulation therapy, in a broad sense, you may think it's this, but there's one big mechanistic difference. I'm not talking about light through my retina and then along the visual system into the brain, as you see here in this diagram. Now, this is an incredibly powerful pathway, an incredibly powerful source on how we feel, how we act, how we think, our biological clocks, how we sleep. And some of these things we could do with photobiomodulation therapy by direct innervation of parts of our body or our brain. But this is not this type of light therapy, as it's normally called. And I'm sure all of you have heard or seen about winter blues or sad seasonal affective disorder, where you don't get enough light, you feel bad, you gain weight, you get depressed. And so, in fact, you could buy, Amazon sells every kind, different wavelengths of light, light therapy boxes that you sit and you don't look at directly, but you have in the room, and you're getting this indirect exposure to light. So that's not exactly what I'm talking about either. And the third thing I'm not talking about is things like photodynamic therapy. Now, I'm sure you've all heard about PDT, photodynamic therapy. People are out there, you're educated people, I know you know this. But the difference here is that we're talking in this case about exogenous chromophores. Now, when I say exogenous chromophores, what I mean is that these are not produced and resident within our body. These are things that are produced out of the body and then put in our body and then shine light on it, and we get an effect. So a chromophore is actually a molecule that has atoms in it, usually a metal, for example, like copper, that can absorb specific wavelengths of light. And that's what we're talking about when I say a chromophore. So in the PDT case, as you see here, you are given a photosensitizer, and then it circulates about the body. And then usually this is used a lot with tumors. It settles within the tumor. And notice that it says idealized because it actually doesn't usually all go there. And then you shine light on that area of a specific wavelength for the photosensitizer that you put in. And voila, the tumor is gone, and our little friend is very happy, as you see. So before I get to the actual definition, there's a couple words, more words I'd like to, and concepts kind of review with you. The first is the electromagnetic spectrum. Now, as you see in this diagram, the electromagnetic spectrum includes all types of electromagnetic radiation. And I think on the bottom panel here, this gives you an idea, and you know, I'm sure, all of these radioactive sources, medical x-rays, sunlight, all the way down here to radio waves and beyond. The other thing I want you to realize is that, we have different frequencies of these wavelengths. And on this end, gamma and cosmic rays, we have very short frequencies or wavelengths. On this end, we have long wavelengths and much lower power. They're not as powerful as you see. And then we have sort of a central zone that we're going to be focusing on tonight, and that's right in this region. That zone is spoken of as optical radiation. And this includes UV light that you go outside and get a nice tan from, and possibly some of these UV rays you don't want so you don't develop skin cancer. We have visible light, and we actually define that as light because our eye can perceive it. And then we have infrared radiation at the far end. So we have this optical radiation that's spanning from 100 nanometers out to 1 millimeter. And what we're talking about tonight in photobiomodulation therapy is from the region of 400 out to 1,000 nanometers. Those are the wavelengths that are important. And in that 1,000 range, actually above 700 to 1,000, those wavelengths we talk about near infrared. They also call them IRA, but you see IRA goes beyond that. So it's just the beginning of this IRA segment. Now, another interesting thing that I want you to remember about that optical radiation is if you look here, you see this region is falling in what we call non-ionizing wavelengths versus ionizing. Now, out at ionizing, those are the real high power areas or wavelengths, as we talked about metal x-rays, where all of the wavelengths we're going to be considering in photobiomodulation therapy are in non-ionizing. And what does that mean? That means they don't have enough energy to remove an atom or molecule. So it doesn't ionize anything, and it doesn't damage the DNA. So finally, after all that introduction, we're at our definition of photobiomodulation. So photobiomodulation is the mechanism by which non-ionizing optical radiation in the visible and near-infrared spectral range is absorbed by endogenous chromophores to elicit photophysical, photochemical events at various biological scales. Now, I told you before, exogenous chromophores were that light-sensitive material that was added to your body. I'm talking about here light-sensitive molecules that are resident within your body. Photobiomodulation therapy is the therapy based on that definition, And one of the really important things that you need to think about is that there's not one type of light that can do this. We have specific wavelengths that we need, but the source of those wavelengths could be lasers, they could be LEDs, or they could be a broadband light source that includes many wavelengths. And again, they have to be in this visible and infrared spectrum to get the physiological changes that you will get health benefits from. Now, as I said, when I started in this field, we didn't really call it photobiomodulation. And the pioneer who really noticed that something was going on was a Dr. Mester from Hungary. And his first publication came out in 1968. And he called what was doing the effect he saw low power laser rays. Now, the first laser that was invented was a ruby laser. That was a red light emitting laser. It was very hard, very, very hard to get high power out of it. But a lot of scientists, it was military driven to see if it could cause cancer or if it could cure cancer, or if it would cause brain injury. So a lot of research was being done, and Dr. Mester got one of these lasers, but I think his power was on the low end, probably lower than he might have thought, because he was seeing if he could affect cancer cells that he injected into a rodent. Now, he didn't do anything to those cancer cells, but he was an observant man, and he noticed that in the region he put the cancer, he had shaved hair, and the hair grew back. Rapidly grew back. And he became interested in it, expanded his research, focusing on wound healing right in the beginning. And he found that using this light, he could accelerate the rate at which these wounds closed in his rodent models. Now, I've done wound healing work. I didn't bring background research on what I've done, but I want you to realize this has been done and published since 1970s, and there's a huge amount of research out there on cutaneous wounds, on chronic ulcers, chronic bed wounds, just about every kind of wound that responds beautifully to this therapy. Now, at this time in the 80s, industry got interested, and they thought, well, I'm going to build a laser. Got a little, we have Heaney lasers now. We have all kind of red light lasers. But we wanted it to be different from surgical lasers, which had swamped the field. And so they came up with names like cold, cool laser, cold, low-level laser, or soft laser. All that was to say, this isn't a surgical laser. And then we came up with two terms, or in the past, they came up with two terms, low-level laser therapy and low-level light therapy. And these really stuck. And so around 2003 to 2005, I was working with the DARPA team on accelerated healing. Believe it or not, they were interested in growing a limb very quickly. And it was a great effort. But my manager at DARPA came to me and the group and said, I can't stand this name, low-level light therapy. It implies low-level research. And DARPA doesn't do low-level research. We're going to change this name. So we had a sort of meeting with some thought leaders, and we decided we would go with photobiomodulation as the mechanistic definition I gave you, and photobiomodulation therapy as the name of the therapy. And so we try to spread the word. People who were in editorial positions as myself, if somebody wrote a paper, we said, please use this term, it's more accurate. And it sort of hobbled along. We did get interest. And then finally, around 2015, myself, Dr. Ray Lanzafam, who actually was the first person to get photobiomodulation therapy in print, that term, and Dr. Arani published this editorial piece saying, from now on, this really needs to be called photobiomodulation therapy, and we gave all the reasons. Now, one of the reasons that people said we don't want to accept it was that if you do a web line search and you put in photobiomodulation therapy, it wouldn't bring up all of what was low-level light references because it didn't have a search term at the National Library of Medicine. But Dr. Irani was working for a few years at NIH, and he made it his effort to work with the National Library of Medicine, and that mesh term, photobiomodulation therapy, was added to the search terms in 2016. So we were ecstatic. It's really spreading. There are still some people who, this doesn't happen in any other field, I don't know why, feel compelled to make up their own name every time they publish a paper. But we'll see. Now, one other slide I want to show you, so you really understand when people talk to you or a company comes to you and says, boy, do I have a laser for you. I want you, and that's kind of how I pitched this talk tonight, I want you to understand the background, I want you to understand the science so that you will know what's the good, the bad, and the ugly in this field right now. So actually, low-level light therapy was coined by two doctors, Dr. Calderhead and Dr. Oshiro, back in the 1980s. And Glenn Calderhead, who's a friend of mine, actually let me use this diagram that they had created. And low-level light therapy actually refers to the level of tissue interaction. And you see in the bottom of this, there's a damage threshold. And below that, we're not getting damage. We're getting cellular photoactivation, we're getting therapeutic reactions, and we're below the damage level. And that's really what that meant. But over the years, it's kind of morphed to mean power. People think low-level laser therapy is low power, okay? And I want you to realize that. Now, I personally was so ecstatic that things were looking up in the terminology world. And about a year ago, papers started showing up, and I was assigned to be their guiding editor with new terms, such as laser photobiomodulation therapy, LED photobiomodulation therapy. Where now companies who made a laser or made an LED, they wanted to make their device something special. So they gave it this name. It's all photobiomodulation therapy because in the definition we have, it doesn't matter on the light source because basically they're all going to use the same wavelength rate. They're all going to use the same output powers that could give the appropriate dosage, and it all has the same basic mechanism. So every time I saw this, I wrote a rebuttal to the authors and essentially made them change it if the paper came to me. Then last week, in the Journal of Photomedicine and Laser Surgery, much to my displeasure, a guest editorial showed up, and the title was Low-Level Laser Therapy and World Association for Laser Therapy Dosage Recommendations. Now, Dr. Irani was instrumental in changing the name of the World Association of Laser Therapy to World Association of Photobiomodulation Therapy a year ago, but this is now ignored. And there are two of these authors are past presidents of WALT. And then the first sentence, and this isn't the complete sentence, said, and I think you should now be able to be aware of how ridiculous this statement is. Low-level laser therapy is a type of photobiomodulation performed by class 3B lasers. So they're basing it all on power output, and they're using these names again as if they're different. This is not a step forward for this field. It adds confusion to patients, it adds confusion to people who want to purchase a laser. So that's enough on that. Let's move on to some mechanism so you understand how this works. So the first law of photobiology, not just with photobiomodulation therapy, states that for light to have an effect on a living biological system, photons must be absorbed by some molecular chromophore. Remember, I told you it's a molecule that has atoms that can absorb a specific wavelength of light. And PBM uses endogenous chromophores that are resident in our body, and it absorbs specific wavelengths of light in that 400 to 1,000. And these exist within our body. And where they exist, the primary photoacceptor is thought of now to exist in mitochondria, in the inner membrane of mitochondria. Now, you know, in biology, there's kind of a rule that a lot of people think about, and they say, why would you have deep in your liver a protein that could absorb light that never sees that light? Because usually, if you have a protein that's not functional anymore, your body gets rid of it, okay? There are two things. In some cases, these proteins have taken on a new function, maybe as a calcium transporter. And the other idea to explain this is to go back to the beginning in the formation of mitochondria within our cells. And about 2 billion years ago, we had different types of cells called prokaryotic cells, aerobic, and we had anaerobic floating around. And what do I mean by prokaryotic? See that little red squiggly line in there? That's the DNA, and it's just floating around inside this organism. They don't have a nucleus. But with time, that DNA is gathered up inside a membrane, and a nucleus is formed. And in this field, they changed the name from prokaryotic to eukaryotic. But these cells were still anaerobic. They couldn't use, or they didn't use oxygen. But there floating around also in the great milieu was these aerobic prokaryotes. And it's believed that the eukaryotes engulfed them, took them into their body, and then a real smart thing it did was take away the majority of its DNA's job and move it into its nuclear job so it could control the mitochondria. But that mitochondria became the energy machine in the cell, and it allowed the cell to use oxygen. And that's what we mean by an endosymbiotic relationship, a living together inside. And it's believed because these things existed before that they gave rise to chloroplasts in plant cells and mitochondria in animal cells and retain this photo-absorbing capacity. So now if we look back at the mitochondria in our current day cell, and you see it had a membrane and then it was engulfed by the other cell and wrapped it in a second membrane. So mitochondria are interesting, they have two membranes. And this protein that will absorb the light is absorbed by copper centers, and the name of the protein is cytochrome C oxidase. And it actually occurs in this complex 4 on the inner membrane, and it's part of the respiratory chain in the mitochondria. So that when red light, as you see here, makes it to the cell, it's absorbed by the copper centers. And if the mitochondria are not functioning optimally, as in many diseases and aging, then you get an increase in the mitochondrial activity, and some other things happen. Oxygen consumption's increased. There's a restoration of these electrochemical gradients across the mitochondria, so it's working more effectively, and you get an increase in ATP production. Now, if you shine a light on a cell that's just happy as a clam, those mitochondria are firing at 100%, and you put light on it, you're not going to get an effect. But the reality is, in many serious diseases, it's a mitochondrial, at least component-based disease. And as we age, unfortunately, our mitochondria slow down. So once again, we have this near-infrared light. We're shining it on our cell. And I told you we get, and these are the 3 things. Now, mitochondria do a lot of things, but most people emphasize these 3 things. Increases in production of ATP, which can act as a signaling molecule inside the cell, or it can be extruded from the cell and affect another cell as a signaling molecule. You also get production of reactive oxygen species, but this is just a small amount, and small amounts of ROS are beneficial to the cell. And that sets up another signaling pathway to the nucleus. And we also believe that we get the expression of NO with the utilization of oxygen, because NO is used by the mitochondria to regulate its oxygen usage. And that leaves the cell and causes vasodilation. So you get better blood flow within the region where the light was. And so all these cellular events can initiate downstream signaling by turning on gene expression and making RNA and new protein. And we'll talk about this some more. Now, I'm going to talk in the next few minutes. I have to keep my eye on the time here. And that's that you can have changes at 3 levels. At the molecular level, such as I'm talking about within the DNA, RNA, or protein, in the signaling systems. At the cell tissue level, and then at the system level. And I'll give you one example of each of these. So once again, just to remind you about these 2 major pathways that are often implicated, and that's the cyclic AMP pathway to protein kinase, and then ROS activating this NF-kappa B pathway, which both of those turn on gene transcription, which is equal to RNA synthesis, and then you get translation of that new RNA and new proteins made. So if we look at, just for one example, this cyclic AMP pathway, you see what happens. We have an increase in cyclic AMP, and that stimulates this protein kinase A. And the protein kinase A makes this happen. So you have this Krebs molecule, which is cyclic AMP response element binding protein. Well, it takes a phosphate away from the ATP, and it adds it to the Krebs. And that activates the Krebs to attach to this part of the DNA called Cre, and that turns on transcription. Now, in terms of photobiomodulation, and one of the big things that it does is moderate inflammatory response in the body, is that there's a Dr. Lima, he's a very good researcher, and he found that PBM could increase cyclic AMP. And he was working with red light. You see 660 nanometers is red light. And wavelength, the typical wavelength around, a typical energy density around 4 joules per centimeter square. And he was looking in animal models in their lungs and airway. And he wanted to look at this anti-inflammatory action. So he already knew from his studies that he could increase cyclic AMP with PBM. And he also knew from previous work that he could upregulate cyclic AMP by such factors as prostaglandin E2. And that would inhibit the synthesis of tumor necrosis factor and downregulate the inflammatory response. Well, Dr. Lima, besides finding that he could increase cyclic AMP, he found he could reduce the tumor necrosis factor in the treated lung tissue. And he hypothesized that this treatment increased the cyclic AMP levels, decreased the TNF-alpha, and downregulate the inflammatory process. No, I'm sorry, that's a lot of details, but I want you to realize that there are scientists who are actually looking at this pathway, and it's not just, woohoo, we can change inflammation. We can change inflammation, but it's by a very specific series of cellular events that PBMT, the therapy, can start. Now, I've been working for a while with stem cells, and I was doing a study with normal human neural progenitor cells. My expertise is nervous system. And we were looking with 810 nanometer light. And so a lot of things that I do in my lab, especially if I'm working with tissue culture, stem cells, is that we always start to try to find the most appropriate dosage of light, because that's the key to this whole field. And so I always start with sort of a grid experiment like you see here. So I have different powers that I deliver to 1 centimeter square area that has cells in it. And I alter the times, which gives me a different amount of energy delivered to that same area. Okay? And then I look at some bioassay, ATP production, proliferation, I don't care, any of them that this is supposed to do. In this case, I knew I could put light on neurons and make their neurite processes grow. We've done it a million times with students in the lab, just for fun. And so we knew this is a wonderful bioassay. And the one thing that we found is that there's not just one combination of time and irradiance. Irradiance is power per an area. But there are several combinations that could do the job. And that's good news because a lot of people will try different combinations, which means that the success rate for this instrumentation is increased. Now, when you grow stem cells or progenitor cells, what you normally do is you have your cells and first you put them in a soup that has a specific amount of types of factors, growth factors, and that causes them to divide. And then you put in different factors, and that causes them to differentiate. And I started thinking at this time from observations I've made on other things in the lab, I wonder if I can get these progenitor cells to differentiate and proliferate just with light. And that's what we did. We did this experiment you see here, where we used typical soup, the factors, and down here are the different kinds of factors that have been added. A control with nothing. This is what these cells look like. Well, they're clumps of progenitor cells. And then we added light. And I used what I found out on that chart to deliver the dose I wanted. And this is what the extension looks like. So this first panel is just drawings in pink to show you how we go in with a microscope and then measure the neurite processes. This is staining nuclei. Here in these 3 panels, we use immunocytochemistry. So we use an immune response to tag the neurites with a specific protein. And in this case, it was called double cortin. And double cortin exists when neurites are outgrowing or neurons are migrating in the developing nervous system. And here you can see, this is what it looks like, control. This is what it looks like with the factors, and this is what it looks like with the light. And we found that the light caused a significant effect equal to all those factors, and there was no difference statistically between the factors and the light. And when we decided to probe this further, we looked at growth factor mRNA expression in these cells, where we treated the cells 3 days with the light. And we were very pleased to see that we got an upregulation at days 3 and 7 of a number of very important, it's not the last panel, very important factors. So essentially the light allowed the cell to turn on, and if it wanted to proliferate or differentiate, it now had the energy to produce what it needed in its factors to do the job. So we got a patent for this work that's been issued in the United States, Europe, and in some parts of Asia. So at the system level, and I really wanted to talk to you about this tonight, I want to talk to you about the work I've been doing the last 2 years on pain control. I don't have time to go through all the experiments. One I'll present sort of a summary form. But based on our work, we have proposed that there are 2 different methods that we could use, and probably they're most effective if we use them together, to modulate pain, to change this chronic pain pattern, and to block pain transmission. And all of it, my lab showed, is based on the dose of light you give. So if you look at this panel, we found, and also based on the literature, that if we're under a irradiance of 100 milliwatts per centimeter squared, that we sort of turn on everything that's generally spoken of in photobiomodulation. First of all, we did in our animal models get decreased pain response. We totally changed the chronic inflammatory pattern. And it was amazing because we caused inflammation by damaging the sciatic nerve. But when you have pain turn on that changes to chronic pain, you get inflammation up your entire ascending sensory system that's maintained. And we found with the treatments we did in this range that we changed that. We started getting expression of anti-inflammatory markers over pro-inflammatory markers. And then we also did some experiments that if we upped the output power and we used 250 milliwatts and above, that we could get a rapid block of pain transmission. And this is utilizing the inhibitory aspects of photobiomodulation. It's just not stimulatory. The dose was high enough that we altered the microtubules inside the neurons, which stopped mitochondrial movement, blocked action potential, and stopped the pain. So we really are proposing, and I must say I'm very pleased because I just received a grant from the Defense Health Administration, 2 years of funding to work on these two methodologies. But we're proposing in cases of severe pain that we can block the pain and then go to the lower levels of treatment to take care of the chronic inflammation and break this pain cycle. So a couple of years back, a young functional neurosurgeon from Brazil did a study, and her name is Dr. Holanda. And what she did was she got patients that she put into 3 groups: an ultrasound group, the standard lidocaine injection for back pain, which these functional neurosurgeons will put in at the lumbar 2 dorsal root ganglion level. And then these Brazilian companies tend to make very low output devices, and you can see here that the average radiated power was only 100 milliwatts, but they put it through a tiny fiber, and out the tip of that fiber, it came out as 35 watts. And she put that fiber down, the same needle she used, same size needle she used to inject lidocaine, and put it not right up against dorsal root ganglion, but envisioned this under the fluoroscope within half a centimeter of it. And this top panel, I'm sorry if you think it's a little gross, but this is down in my university's cadaver lab. I teach medical students head and neck anatomy and neuroanatomy, and so I have access to the material, but here's the dorsal root ganglion. And we took our device and we measured what was actually hitting that ganglion, and it was about 4 watts. It was probably still a lot higher than she needed. But the interesting thing is the pain. She could ask her patients, what are you feeling? The pain stopped within 5 minutes, and the pain was suppressed for 21 days in these patients. She's now trying to get funding to do a longer study. The interesting thing that the pain blockage was comparable in lidocaine and comparable with the light through this fiber, but they were both much better than ultrasound. I know a lot of people are running to get ultrasound for their back pain now. So she came to my lab then that fall, and we wanted to look at the mechanistic basis of what was happening. And so here is a pretty cell, dorsal root ganglion neuron in culture. And an Australian investigator, Dr. Roberta Chow, was working on also dorsal root ganglion, and she published using a different wavelength that in culture, if she used 300 milliwatts per a centimeter square area, which is high. Usually you're like less than a milliwatt, percentage of a milliwatt when you deal with cells, that she could get different changes in the mitochondria in her cells. And we did that, and we looked at mitochondrial metabolic activity, and then we looked at the neurons themselves and what happened to them. And so in this panel, you could see here, using this MTS assay, which looks at metabolic mitochondrial activity, that we downregulated it at all these different times. And I don't know if you could see it with the lighting, but at all these panels, these neurites that were growing out from the ganglion neurons form these bowls, like balls on them, like rosary beads, and they're called varicosities. And it's known that these varicosities are due to breaking of the microtubules. And so the functional change then, and the other interesting thing that we saw was that only the smaller neurons, because you get a dorsal root ganglion, it's different sized neurons in it because these neurons do different jobs in your body. And the smaller ones, 30 microns or less, are the ones that transmit pain and temperature. They're the ones that were affected. The big fibers, which are proprioceptive, allow you to know where your body is in space and to walk correctly, they weren't affected. And that's incredibly important, because if you want to do a treatment and block pain, you want that person to keep their motor function. And in fact, that's one of the things we're doing with the military funding, is to see in a mixed nerve, not just sensory nerve, if we can block pain and maintain motor function, which we think is very feasible because motor neurons are the largest in our bodies. So no one had done this before, and I wanted to look, and there's a point to this slide besides my measuring light transmission, although I do measure light transmission all the time. But my lab was the first to measure light transmission in anesthetized rodents. Before this, people used dead rodents, or they used Monte Carlo mathematical modeling, or they used created phantoms like fake tissue. But we used anesthetized rodents. And Dr. Sean Wang built me a special sensor where I could actually measure irradiance per area inside the animal. And I wanted to see, I had an 810 nanometer laser that I wanted to use, near infrared. And I wanted to see, what would I have to put on the surface to get near 300 milliwatts inside the animal? Now, we're talking here, folks, about an animal with no pigment. These are albino. There's no melanin to absorb light. And look at that. It's not a big distance between the nerve that I was measuring and what light got to it. We're talking maybe a centimeter to 2 of tissue. I needed to put 10 watts of light to get 270 milliwatts to that nerve. Now, I'm telling you that because in the olden days, when this field started and it was red light and people were doing wounds, that was surface, it didn't matter. But then people started saying, well, let's do deep tissue. Let's do the stellate ganglion and see what happens. And you know what happened? Nothing. Because on the surface, they put the dose they were using in the tissue culture or for wound closure. And they didn't think that 30% to 40% of that light will be lost just passing through the dermis. So if you want to get light inside, you have to put a lot more light on the outside if you're doing transcutaneous application. And that's a really important point. I want to make sure we have time for questions, so I'll speed up here. So what we did was I did a spared nerve injury model, which is a very classic way to create a pain model in a rodent. You take the sciatic nerve and you cut it, and then you ligate it, and you leave one intact. And you get this big inflammatory response within the dorsal root ganglion, which makes the axons that are in the sural nerve sensitive. So now they start acting as if they're experiencing neuropathic pain. And then what we did was we took these rats, you had to wait 7 days, and we tested them. We did all kind of behavioral testing to make sure that they were acting now with heightened sensitivity to stimuli. And at that point, we did this test, and we took that 10 watts we knew we could deliver 270 milliwatts inside, and for 2 minutes we ran it from dorsal root ganglion along the nerve out to the cutaneous region. Now, we couldn't ask our rats, hey, how does that feel? We had to wait for them to wake up. So we didn't get a 5-minute response. We got an hour later response. When they woke up, we did the testing. And then we also did some long-term experiments. And the beauty of it was that for the pain, pinprick test, the heat test, the cold test, that we could within an hour change this, and it was maintained for various amounts of time depending on what that sensation was. So, there's room for more research here. But the really interesting thing, just like in the tissue culture dish, we didn't affect the big neurons. So again, I thought that was just, every now and then an experiment works just like you think it does the first time, and it is the best feeling in the world. So every time I look at that, I'm just so happy. So basically, our preclinical work in animals and tissue culture shows that we could do dosing to affect pain and inflammation related to pain by doing different dosing. And I think the most effective therapy will be to combine those. And that's basically what these slides reiterate for you. Now, what if you have back pain, or maybe you have migraines or a pulled tendon, and you want to go get treated? So in the next couple of minutes, I'm going to tell you some of the things you need to think about, and make sure that if you buy your own device, you're thinking about these things. Or if you go to your clinician, you say to him, what's the wavelength of that laser? Maybe they'll say, I don't know. And then you walk out the door. Or maybe they'll say, oh, well, this is a dual wavelength, and you'll say, okay, I'll stay. So you've got to think about what you want treated and where it is in your body. If it's a wound, you can use most wavelengths if the dosing's right. And in fact, red light has been shown now for 50 years to do an incredible job on healing and ulcers and burns. So it's very easy to treat, and it's very effective. But what if you have that really bad back pain? You've got to think about the wavelength and how deep it goes. It doesn't usually go this deep. I tried, but... So it's wavelength that we have to consider. And then the other thing is you got to get light where you want it, and you got to get the right dose. If not enough photons get there, it'll be suboptimal, and you're not going to be fixed. So the goal in deeper tissue PBM is to look at all these wavelengths and look what's waiting for that wavelength of light. Water, melanin in the skin, blood, oxygenated and deoxygenated. And so all these things have their favorite wavelengths to absorb. Oops, sorry. I keep pressing it instead of the... All of these things have their favorite wavelengths, and you can see their extents. Here's melanin, and you could see that just beyond melanin, there's a 1064 nanometer. I haven't worked with that much, and I'm dying to work with that wavelength because it's just beyond melanin. Water absorption is still pretty minimal, and I think it would be a very effective clinical wavelength. But if you're treating, or you are a person of color, you want to be aware that one, certain wavelengths could burn your skin because of the melanin absorption, but also the treatment would have to be different because of the melanin absorption. Because a famous guy in light tissue interaction, Dr. Stephen Jocks, has shown that if you use red light, it's absorbed 4 times more than an infrared by melanin. Okay? So these are all other considerations. So this is my famous rat. It's all over the web. I don't know how it got out there, but It's everywhere, and I go to talks and people show it and use it in a totally wrong context, but that's okay. So we did this experiment with my colleagues from the FDA, Dr. Ronald Wayne and Ilko Elef, and they're physicists and they were optical fiber experts. And you could see here, this is the first anesthetized rat we ever studied, and we put white light on the rat's back, and then we had an optical fiber that we could insert below the skin, into superficial fascia, into muscle, into bone, into the vertebral column, and into the CNS. And the most important thing here is we found that in the lowest layer, all the way down to the vertebrae, that there is a window between 790 to 820 with a peak at 810 nanometers that has the deepest penetration into the body. And again, our work on spinal cord injury using this wavelength has resulted in issuance of a patent for this use. The other beautiful thing we found also using live animals, and we looked at blood transmission, 810 is minimally absorbed by blood because it's right where you have oxygenated and deoxygenated blood profiles, absorption profiles cross. And also, it is minimally absorbed by water. So it's a great wavelength if you want to get inside. Now, if you use 808, that's not going to make any difference. Some people use 820, 830. A lot of that has to do with getting around patent on what they're trying to do. I want to mention to you about transcranial laser therapy. I think I'm going over my time. Shall I just finish with this? So, okay, I'm just going to tell you about this transcranial laser therapy so you know that one of the real hot areas right now is the use of light through the scalp, through the skin, I'm so klutzy, and then into the brain. And this is really important. And we tried to get some funding from like the Michael J. Fox Foundation, and they came back and said, look, no one's proven you get light into the brain. Go prove that and come back and ask us for money. And it's really a very exciting field because people in preclinical work and in some small patient studies are showing great effects on Alzheimer's, some effects on Parkinson's. Parkinson's is trickier, but great effects on post-traumatic disorder, and on other types of brain trauma. So it's got great promise. I'm on the safety and efficacy board for a study that's going on in New York and Boston right now, looking at this light for major depression that's non-responsive to drugs. And I'm so anxious to see when that data is unveiled, and that's got a good patient population. So in this study, we started to go ahead and look at the light. In this case, we just looked at brain. This is fresh human brains that we got through the state of Maryland cadaver program. And you could see if we this was 810 nanometer light, that the light is transmitted into the brain. And you know how the brain has these bumps and grooves, gyri and sulci? Well, look what happens. We found out that the sulci essentially act as light waveguides, and the light could get, I told you, deeper into the brain. So we went on and we did a much more elegant experiment. This was led by Dr. Clark Tedford, who is currently got fabulous data on the use of light for dry macular degeneration. Really wonderful data. And he came up with this rig where we could cut off the top of a cadaver head, fresh cadaver, cut the head in half, mount it here. We had a laser on the outside, and then these are all sensors. And basically, the bottom line is in this panel that we found that light could get 40 millimeters or 4 centimeters into the brain. Now, whether this was the optical, is a truly photonic optimal dose isn't known. Now, I want to end, but I just want to tell you the other thing using this slide is that wavelength exclusively determines depth of penetration. Its wavelength, don't let anybody tell you anything else. It's not pulsing, it won't change it. The output power won't make it go deeper. All of that is fantasy. What you do get when you increase the output power are more photons along that given depth. And that allows you to regulate the dose you want to get to these deep tissues. There's a bunch of more studies. Yeah, I could go on forever. But I'm going to go to the conclusion, and hopefully you have any burning questions. I think that this photonic medicine is the future. And I think because of its mitochondrial mechanism and mitochondrial involvement in so many diseases, I think that it could address a lot of diseases we don't have a current cure for. I think that there is extraordinarily strong preclinical data. There's some indication in small clinical data, but we need a big trial to really be able to say there's strong data there. It's non-pharmaceutical without those side effects, and it's non-addicting, and it offers a wonderful opportunity for pain control. And also another hot issue, I've just been invited to actually attend an LED conference in France in November, where they are focusing on the use of LEDs in the aging population, which is now huge. Both body function and keeping your muscles strong and responsive, and continuing to build muscle instead of lose it, also with light, and preventing or slowing cognitive decline with aging. So those are all wonderful things. The last thing I'll say is there are a lot of shysters out there right now. You can see how people are trying to manipulate even the language. But you can find all kinds of devices, and a lot of them are really cheap LEDs that don't consider these things. But there are excellent companies who are investing in research, who are investing in scientists who are doing mathematical modeling and measurements and can make wonderful dosage recommendations. So I hope that tonight I just gave you some of the things I want you to think about and realize that this is a wonderful therapy, and don't get caught up by the shysters. You know what to ask them now, right? Thank you for coming. I appreciate it. So I know that the president has a reception that you're probably all dying to get to, but if anyone, I'm glad to talk to you all out there about anything, but if anybody has a real burning question now, I'd love to answer it. Yeah, they're recording this, so we'll need.
Since PBM is being used throughout the world, why is the U.S. behind? Because you go to Canada, a lot of PBM treatments are now being covered and reimbursed in Canadian Health. National Health Service in England is now approved and doing reimbursement throughout the European Union, Australia, New Zealand, Brazil. Why is the U.S. behind?
You know, I gave a talk at the Optical Society of America, and I looked into this non-acceptance. Part of it was this confusion in terminology. It was like the community just couldn't get together. They couldn't get their act together. But more importantly, it's a historical reason. And that's because that first investigator, Dr. Mester, was from an Eastern Bloc country, Hungary. And this was the Cold War days. And he published his stuff, and people would not accept anything that came out of the Eastern Bloc. And essentially, if Americans didn't prove it, it didn't exist, and it wasn't right. And that started to set the negative thinking in this country. And people still believed in it, but it became this sort of fringe alternative, and it wasn't mainstreamed medicine. So some physical therapists, chiropractors, mystics were dabbling in it. And so it became a wall of prejudice against it for the mainstream medical community. And I think it's taken 50 years and maybe shorter now that Scott Faulkner's on the job. I think he's done more in 6 months than I've seen get done in policy and the word out to the government in 40 years. So I congratulate you on that. And I think things like the opioid crisis, which is so incredible across this whole country, has now got to be addressed, and people are starting to listen. So I'm hopeful. Yes. Yes.
So, well, this is just this is an editorial comment, and Juanita can disagree with me if she'd.
I disagree.
If she'd like to. But if we had a pill, if we had a product that was made by a pharmaceutical, multi-billion-dollar pharmaceutical company, with the wherewithal financially and infrastructure-wise to push this forward, all the things that photobiomodulation would do, it would far exceed the sales for Prozac and Viagra combined. So I think that that's part of it, is that the laser manufacturers are all small companies. Nobody's got $1 billion to do the necessary paperwork for the FDA.
I agree. Any other question?
Juanita, thank you so much.
You're welcome.
Wonderful.